Right. Hello, everyone, and welcome to our final lecture within our um, second module within our Agri-informatics lecture series. The final lecture of this module will, of course, again be delivered by Professor Bruce Walsh, who's a professor at the University of Arizona. Since today's lecture is going to be a little bit lengthy, we won't spend too much time doing introductions again. We have introduced Bruce a couple of times before, so I'll just hand straight over to Bruce and you can kick off.
So welcome back, everyone. Again, I want to emphasize, as we've done through all the other lectures, there'll be a fair amount of kind of mathematical detail in this lecture. Don't worry too much about that. That's stuff you look up if you need it. Um, and hang on, sorry, there we are. And so don't try to worry about following the math or anything of that nature. Just try to get the general feel and the general concept, because as with other lectures, you have notes to go with this. And in particular, again, this is a slide we've had before. Well, what you have now are chapters 34, 37, and 38. Those actually are not yet published in any book. Those are chapters going to come out of the second part. So Walsh, Fisher, Lynch, 2025 is the first part of the revision of this. The second part will have these, and these are on multi-trait selection, index selection theory, and index selection applications. And these have a huge amount of material in them. So multiple traits. When you go from selecting on a single trait to multiple traits, very interesting things happen. And so the biggest takeaway is you cannot simply take the breeder's equation when you're selecting on two traits and apply it separately to each trait. You're going to be fundamentally wrong, and I'll try to show you why that's the case. So to start with, we need to have two different types of correlations that we deal with: phenotypic correlations and genetic correlations. So within an individual, trait values can be positively or negatively correlated. For example, height and weight, positively correlated; weight and lifespan could be negatively correlated. Um, these are phenotypic correlations. I can measure them directly. For example, I can measure plant height and plant biomass. So phenotypic correlations are important when we talk about how means change within a generation, that is, that S we saw before. To translate a within generation change in mean across generations, we're going to need genetic correlations. So here's basically how to think about it. The arrowheads have been missing in the X's. Don't worry about that. So here, this is in an individual. This is in you. Imagine X is your ear size and Y is your nose size. There's a phenotypic correlation. If I take the entire class and plot for each individual ear size versus nose size, I get a scatter plot. It probably has a positive slope. So that's phenotypic correlation you observe. That has two components. You could have correlations among breeding values. So, for example, it might be the case if you have a high correlation for ear size, a high breeding value for ear size, you have a high breeding value for nose length, or it could be negative. That's the genetic correlation. And that could either be based upon breeding values. Remember, breeding values are what a parent passes on to its offspring if you cross them. Whereas these could be genotypic values if we talk about offspring being exact clones of their parents. So there's a phenotypic correlation which results from a genetic correlation and an environmental correlation. And the key here is that correlation in breeding values, again, we'll typically use that when we talk about genetic correlation, but again, it can also apply if offsprings are clones, in which case instead of the breeding value would be the genotypic value. Now, these correlations can arise from pleiotropic effects. For example, a gene that affects biomass might also affect overall size of the plant. They can also arise from linkage to disequilibrium. On a chromosome, you may have an allele that gives you higher plant yield, and next to it, you have an allele that gives you lower biomass. Well, if those are statistically associated, there's linkage to disequilibrium, that will also generate genetic correlation. That correlation will decay over time, whereas the only way to break up a pleiotropic correlation is through new mutations that modify the effects. Then we've got what's called a correlation on environmental values, which is kind of everything else. It's environment, but it's also parts of the genetics you can't pass on. And you can put all those together and write it this way. Your phenotypic correlation is a function of your genetic and your environmental correlation. And the weights here depend upon the heritabilities. Heritability of trait X, heritability of trait Y. So if traits X and Y are both highly heritable, that term is large and that term is small. So phenotypic correlations largely track genetic correlations. If heritability is low, that term is small, that term is near one. Phenotypic correlations largely track environmental correlations. With intermediate heritabilities, you really can't say. So your phenotypic correlation is not necessarily a good predictor of your genetic correlation, and in fact may often be a rather poor predictor. And in fact, now that I'm thinking about it, after class, I'll also post the chapter from Walsh, Fisher, Lynch on correlated characters. I've got that written. I'll have that published. I'll post that online for you guys. So an obvious question is, I can estimate a phenotypic correlation by simply measuring phenotypes and doing standard statistics. How do I estimate a genetic correlation? And it turns out it's actually really easy. So recall we talked about the correlation among relatives. So the correlation between a value in the parent and a value in its offspring, that correlation is 1/2 the additive variation. Now recall for a regression, the slope of that regression is the correlation between the two variables divided by the variance in your predictor. So the slope of this regression, I can write it this way, and since I can measure phenotypic variance, I can estimate the genetic variance is simply twice the slope of this regression times the phenotypic variance. So that's a very easy way using regressions to basically estimate the genetic variance of, for example, trait X. Well, what about the genetic covariation between trait X and trait Y? If you think about it, it's also easy. Now what I do is I measure X in the parent and then Y in the offspring. If I measure X in the parent and Y in the offspring, this correlation now becomes the genetic correlation between trait X and trait Y times 1/2, that's the parent offspring part. So now instead of regressing X on X, I now regress X on Y and use the same expression, and now I can estimate the genetic covariance. If I have a covariance, the correlation is just the covariance divided by the square root of the two variances. So I can do X in parent, Y in offspring. Since I have the data, I can also reverse that, do Y in parent, X in offspring. I can take the average of those two, and then there's my estimator of genetic covariance. Don't worry about the details. The key feature here is before we've estimated genetic variances by measuring the same trait in two relatives and then having a weighing factor, twice the coefficient of coancestry. To estimate genetic covariances, I simply measure trait X in one relative and trait Y in another. So for example, if I measure X in the offspring and Y in the parent, that gives me an estimate of 1/2 times the genetic correlation between those two traits. If I measure X in the parent and X in the offspring, that gives me covariance with itself as a variance. That gives me a measure of the additive variance in trait X, Y parent, Y offspring, additive variance in trait Y. I can do the same thing, for example, for full SIPs. More generally, if I measure X in I and Y in J, so X and Y are different traits, so the term that comes in is the covariation and breeding values between those two traits, and this term here is twice the coefficient of coancestry. So we've seen that before, right? That's an eighth with half sibs, and it's a quarter with full sibs. We saw that, for example, from, I think it was lecture 2, we talked about that. So the bottom line is it's very straightforward to estimate these genetic covariances. You simply need to take phenotypic measurements in sets of known relatives. That's a phenotypic measurement, and that tells you how the relatives are related. You can either use a pedigree or marker to do that. But the basic idea is basically measure X in one relative and Y in the other. And as we'll see, that turns out to be very important when we talk about G by E. G by E is very closely related to correlated characters, as we'll come back and mention. So now that we have phenotypic correlations and genetic correlations in mind, let's look at a simple example. So here I've got X and Y, and this red circle here is the scatter plot of individuals for X and Y. And what you see is they're phenotypically correlated. If you're large in X, you tend to be large in Y. And now let's go in and just select on X. So this plus here is the mean of the overall distribution. If I take individuals whose X value is above this threshold, then I have this little wedge. The mean of that wedge is now here. So what you see is that the mean of X in the selected portion is larger than the overall mean of X. So I have a selection differential on X. But I also have a differential on Y. Y was originally here in this little wedge because I've got phenotypic correlations. Y, the mean value of Y in this wedge is larger than the mean value overall. So I have a differential on Y. So what you might think is the response in X is simply H squared of X, the heritability of X times S of X. That is correct. And you might also think the response in Y is H squared Y, SY. That is wrong. That response could be positive, negative, or zero. And the reason it works on X is that I've directly selected on X. So if I select on a single character, the Breeder's equation holds. The reason it doesn't work on Y is I haven't selected on Y. I've selected on X, and Y comes along as a correlated response. Here, that correlation is because X and Y are phenotypically correlated. But does that translate into a change in the breeding values? Remember, that's the key to selection. If I have a group of parents, when I choose that group of parents, does that change the breeding value relative to the overall population? If it does, and those parents have a larger breeding value, then the offspring will be larger. So if this wedge has a larger breeding value for trait X, does it have a larger breeding value for trait Y? Well, if it does, Y will increase. If it's got a lower breeding value, so an average breeding value is zero, if the breeding value for Y turns out to be negative here, Y is going to decrease. If it doesn't change the breeding values at all, Y is not going to change. So when you select directly on a single character, the Breeder's equation holds. When you look at a correlated response, you have to look at genetic correlations. And we'll talk about that now. And again, a lot of these ideas are a little hard to wrap your head around initially. So don't worry too much about the details. Think about the logic and some of the ideas behind it. Then you can go back and read the notes and it'll make hopefully a bit more sense. So now I've replaced that ellipse with simply dots. These are actual individuals. And the red are the individuals that I've saved, and the black are the individuals that I haven't saved. So there's the overall mean for trait X and trait Y beforehand. And if you take the mean in this wedge here, so there's the new mean. So there's a change in trait X, so differential. There's a change in trait Y, differential. That's a within generation change. The key line is for there to be a between generation change. The breeding values must be correlated. What does that mean? When I pick individuals with a large X value, that should tell me something about what their breeding value is for Y. So if individuals with a large X value also tend to have a large breeding value for Y, Y is going to increase. If individuals with a large breeding value for X tend to have a negative, that is a small breeding value for Y, Y is going to decrease. If individuals with a large breeding value for X tell me nothing about the breeding values in Y, they don't change, Y is not going to change. So let's represent that. So in this figure here, each point here, that's an individual, and then the arrows which go from red, would go from black and red, which are the phenotypes, to green and blue. So that individual here, that's its phenotypic value for X and Y. That's its breeding value for X and Y. So now let's look at that slide. So this slide is on phenotypes. If I knew the breeding values, that's the slide on breeding values. The blue are the wedge that I selected. There's the mean beforehand in X and Y. So there's a clearly change in the breeding value of X. I get a response in X, but look at Y. The mean of Y in this group afterwards is the same as before. So in this case, there's no genetic correlation. When I've selected on Y, it doesn't, when I've selected on X, it doesn't change the breeding value in Y. And that's a key idea. When I select on a trait, it only changes another trait if selection on trait X changes the breeding value of trait Y. That is, within an individual, the breeding values for trait X and trait Y are correlated, positively or negatively. So we can kind of put all that together. So here's the regression of breeding values. So what is the predicted breeding value in trait Y given I know the breeding value in trait X. Well, that's a regression slope. There's the regression slope. You can plug it in and get this expression here. So the sign, whether the sign of breeding value of X matches the sign of breeding value of Y depends upon the genetic correlation. If that is zero, the breeding value in Y doesn't change no matter what you do to X. If that is positive, when breeding value in X goes up, breeding value of Y goes up. If that's negative, when breeding value of X goes up, breeding value of Y goes down. What that means is when it's negative, individuals with a large breeding value in X tend to have a small breeding value in Y. And I can put that together and I can talk about this notation here. So R of X, that means the direct response. So R is response I've selected on X. CR of Y means I haven't selected on Y. It's a correlated response. What's the response in Y? And I can write that response in this expression here. And as you see, it depends upon the genetic correlation. And we can rearrange that and do some rearrangement stuff, and we can get an expression down here. Just do one more. Again, don't worry about the details. You can look them up. So here's the expression here. So the correlated response in Y depends upon the strength of selection on X and this term here. X must be heritable, Y must be heritable. But here's the key, a genetic correlation. And this term here is often called, in green, is often called the coheritability, because if X equals Y, that becomes a 1. This simply reduces to H squared X, the normal heritability. So that's a kind of complicated expression that tells me if I select on trait X, what happens on trait Y. And that's for the simplest case of selecting on a single trait and asking how another trait will respond. We'll talk about the general case in a second, which actually turns out to be very easy to write in matrix form. So a really interesting result comes from that. Suppose I have a trait I'm trying to increase. I'm trying to increase trait X. Is there some trait out there, Y, that if I select on Y, I get a larger response than X than if I select on X directly itself? That may not seem to make sense, but let me give you an actual example. So imagine you're looking at fungal resistant in wheat. Now, the head of wheat, think of your fingers sticking out. In the head of wheat, you've got little pockets between your fingers. A fungal spore has to get in that pocket in order to have an infection. So I could measure resistance by planting plants, spraying them with fungi. That's complicated, it's messy. And then select on those plants which show resistance. Or I can measure the gap between your fingers. If that gap is small, it's hard to slide that little marble of a spore in between your fingers. And sort of instead of directly selecting on fungal resistance directly, I could select on gap size between the heads of a seed, which is actually very easy to measure. I can measure that quickly. If I can measure that quickly, I can put much higher selection on it. I can measure very accurately, have a higher heritability. And so this is the ratio of the response selecting on Y in X versus directly selecting on X. And here's what you get. When this equation is satisfied, you get a large response in X by selecting on Y. Now, when could this be satisfied? Well, it's easier to put selection on Y so you can have more intensity. Y has higher heritability, it's easy to measure than X. And most importantly, the genetic correlation. So if Y, you can measure it quickly, which means you can have more selection because you can screen more individuals. So instead of saving the upper 20%, you can save the upper 1%. If it's got a higher heritability, then as long as this term is moderate, the indirect response gives you a larger response than the direct response. Now, this has profound implications in a number of areas. One way is G by E. Correlated characters is the key that picks the lock of G by E problems. G by E is often thought to be mystical and mysterious and complicated, but it's actually really straightforward. If I have a series of maize lines and I measure them all in Uganda, and then I measure all of them in, let's say, Djibouti, even though it's the same individuals, they're probably subtly different traits. I'm measuring the same thing, but probably different genes are involved and different genes are expressed. So when you have two or more discrete environments, Faulkner was the one that suggested you treat those traits as correlated characters. So wet and dry environments, or I measure it in 5 locations. If the genetic correlation is perfect across environments and the spread among values of the genetic variation doesn't change, there's no G by E. But the correlation is less than 1, or the genetic variation changes among that set as you go across environments, you have G by E. So G by E is a multiple traits problem. And that's the way to handle G by E. I mean, there are other ways, but that's one of the simplest ways to handle. And a good example of that, tying in with the previous slide, is this model that the CG centers have. And in the CG center, you're screening crops. So in a CG center, you have lots of resources. So why is the environment of the CG center? You have very good experimental design, as Rika's talked about. You can have very good control over increasing the heritability. You've got large plots, you can put strong selection. And your concern is, if I select an environment Y, how good is that at predicting what's going to happen in environment X? It all depends upon this genetic correlation. What's the correlation between a trait measured in environment Y and a trait measured in environment X? If this correlation is high, the CG model works. If that correlation is low, the CG model can break down, in that it may be much easier to do sloppier experiments in environments which are much closer, as opposed to elegant experiments. So sloppy experiment, that number is small and that number is small. In an elegant experiment, that number is large because you can replicate. That number is large because you've got lots of resources. But all that elegance is lost if this correlation is small. Okay. Let's take a quick break now and remind people what we've talked about so far. What we've talked about is the notion of genetic correlations and phenotypic correlations. Genetic correlations you can directly measure. A phenotypic correlation is something that, a phenotypic correlation is something you can directly measure. A genetic correlation is something you have to look at sets of known relatives. Phenotypic correlations tell you how within generation changes occur. If I choose parents large for trait 1, are those parents also large or small for trait 2? That depends upon phenotypic correlations. Genetic correlations, the response says, if I pick individuals which are large for trait 1, what does that say about their breeding values for trait 2? If large values for trait 1 have large breeding values for trait 2 will increase, a correlated response. If individuals that have a large value for trait 1 have a negative that is a small breeding value for trait 2 decreases. If individuals with a large breeding value for trait 1, a large phenotypic value for trait 1, have no connection with the breeding value, trait Y doesn't change. So we talked about how you can look at that in the simple case of selecting on one trait and asking about a correlated response. We're now going to go back and talk about what happens with multiple traits. This is where matrices come in. But before I do, let me see if people have any quick questions. Again, the easiest thing is to use that raise hand button. I'll check the chat right now if there's anything in there. And the chat, no, mainly the chat is about the interpretation reading. Yeah. So any questions before I move on to the multivariate version of this? And I'll stop after that and see if people have questions as well. Now, the good news is you might think, given how complicated expressions like this look, right? That's a complicated expression. And that's for what appears to be a simple case. I know I select on X, what happens to Y? Well, what happens when I have a whole bunch of things that I don't know how I selected? I just look at changes. How can I interpret those? The answer is very simple. It's the multivariate Breeder's equation. So now instead of measuring a single trait, I have a list or a vector of traits. Trait 1, seed size, trait 2, biomass, trait 3, root density, trait 4, root depth. So I have a list of things that I've measured. And what I can do is I can look at my entire population. Then I can look at those individuals I've chosen as parents. I can look at the mean of trait 1 and the chosen parents minus the overall mean. That's the first element in this vector S, the within generation change in trait 1. Do that for trait 2, trait 3, trait 4. What we want to ask is, how does that correspond to a vector of changes. How much does trait 1 change in the offspring? That's the first element in this vector R. What about the second trait? What about the third trait? Well, for a single trait, we have this very simple relationship, the Breeder's equation. H squared times the within generation change is the between generation change. The multivariate version is beautifully elegant. There it is. R equals G P inverse S. We'll talk about G and P in a second. They're covariance matrices. G of genetics, P of phenotypic. Does this make sense? Well, for a single trait, G becomes a single element VA. P becomes V. P1 over that is that. So we do recover the Breeder's equation if this is a single trait. This is often written by taking P inverse S and calling that beta. Because what that does is it tells you what traits are directly under selection. So beta removes the changes in means caused by phenotypic correlation. So for example, I have two traits. Let's say I've got flowers of bumblebees visiting flowers. And large flowers have more nectar. And you see that bumblebees visit flowers which are larger and have more nectar. Well, are bumblebees coming to large flowers? Are bumblebees coming to large flowers because they have more nectar? If I know the phenotypic covariances among those, I can compute this expression and get a beta. So beta 1 is direct selection on flower size. Beta 2 is direct selection on nectar volume. If it turns out that beta has a 0 in the first term, it means bees are entirely ignoring flower size, and they're just coming to flowers with more nectar. And so beta, if you simply see the within generation changes, beta is a way to decouple and actually ask about the targets of selection. And written that way, this response becomes R equals G beta. That's often something called the Landy equation. Comes out of evolution after Russ Landy. And we'll talk more about that in a second. So what exactly is going on here? S is this vector or list. Within generation change in trait 1, within generation change in trait 2, within generation change in trait N. And now you have no idea about what the actual targets of selection are. I just give you these and don't say anything else. R is the expected change in the offspring in trait 1, expected change in the offspring in trait 2, expected change in the offspring in trait N. What connects these are these phenotypic and genetic variances and covariances. So for example, with two traits, here's the phenotypic variance covariance matrix, phenotypic variance of trait 1, phenotypic variance of trait 2, covariation between 1 and 2. The genetic variance of trait 1, genetic variance of trait 2, genetic covariance between trait 1s and trait 2. So remember I said that beta is the direct selection on a trait. I can reverse that and show that S equals P beta. So if I see a change in trait J, how could that arise? It could arise because I have direct selection on trait J. Beta measures direct selection on trait J. And it can also arise because I have selection on trait I, and trait I and J are phenotypically correlated. So the bottom line is the sign of this. Trait 3 increases by 5 units. That doesn't tell you anything about the actual amount of selection on trait 3. It tells you about this entire expression. And by computing P inverse times S, you can then compute what these betas are. I'll give an example of that in a second. Likewise, so here I've got phenotypes, variances, and covariances. Here I have genetics. So that's the genetic variance, and that's the genetic covariance. And again, what's the response in trait J? Direct selection on trait J times the amount of genetic variation, plus selection on trait I, where trait I and trait J are genetically correlated. So this could be positive, but this term overall here could be sufficiently negative that even though I've selected to increase trait J actually goes down. So I have a direct response and a correlated response. If I switch those to phenotypes, I've got a direct within generation change and a correlated within generation change. So an example helps. This is data on soybeans. Trait 1 is oil, trait 2 is protein, trait 3 is yield. So for example, here are the variants of trait 1, trait 2, trait 3 phenotypic. They're the phenotypic correlation between 1 and 2, 1 and 3, 2 and 3. Here are the genetic variances, trait 1, trait 2, trait 3. Genetic covariances 1 and 2, 1 and 3, 2 and 3. So what's the heritability of trait 1? Genetic variance divided by phenotypic variance, about 0.5. What about trait 2? Same thing. That divided by that, about 0.27. What about trait 3? That divided by that. So the heritabilities are about, 0.45, between 0.3 and 0.45 for these traits. Suppose now I observe the following. I have a set of chosen parents, and in those chosen parents, they have a decrease relative to the overall mean of 10 in oil, an increase of 10 in protein, an increase of 100 in yield. Two obvious questions. What was actually selected on, and what's the response? Well, if you do the Breeder's equation, what do you expect? Well, Breeder's equation is S times H squared. So you expect minus 4.5 for oiled, 2.7 for protein, 35 for yield. We go through, this is putting in P and G in R, that's putting in S, and that's the command that basically says G times the inverse of P times S. And what do I get? Over here, the prediction from the Breeder's equation. 4.5 unit decrease, 2.7 unit increase, 35.3 increase. What do I actually see? 14, 12, 65. Remember, my S here was only minus 10. What I see is minus 14, just about. How does that arise? It arises because all these phenotypic and genetic covariances mean it's extremely difficult to predict response by just simply looking at a single trait. All these phenotypic and genetic correlations really conflate things, and the Breeder's equation is what basically allows you to decouple those. So in particular, what's the actual targets of selection? So here's the selection that I see, and this is basically standardized for phenotypic standard deviations. So what I see is I have decreased selection in trait 1 and increased selection, slight and strong, in traits 2 and 3. What's actually occurring is I'm directly selecting to decrease trait 2 as well. But I don't see that. Why? Because this other term here overpowers it. So even though that is negative for trait 1 and trait 2, I get a negative for trait 1, but trait 2 I get positive because this negative value is overpowered by this positive value over here. So this example shows you how when you've got more than one trait, you've got to use the multivariate Breeder's equation because you'll have very biased views about the nature of response, very incorrect views about the nature of both a cross-generation response and also about the nature of selection. Let me just make a couple of points and we'll be finished with multiple trait. We'll go on to index. So suppose I see the following. Here's my phenotypic correlations, my phenotypic variances and covariances, genetic variances and covariances. So this is what I see. There's the actual nature of selection. So it's actually 4 times stronger on trait 1. What's the actual response going to be? Well, so beta is the direction you're trying to move. That's as a breeder, that's where you selected for. You're trying to go that way. That's what beta is. What's the actual response? It's this. So I'm trying to increase trait 1. I do increase trait 1, but I'm also trying to decrease trait 2. But here I end up actually increasing trait 2. So my optimal direction, beta, I actually rotate off that a little bit. And why do I rotate off there? It's the nature of the genetic variances and covariances in here. And we call that rotation a genetic constraint. Here's an extreme example of that. So here's the genetic variances. Here are the betas. If I look at one character at a time, I'd expect a 20 unit increase in trait 1 and a 40 unit decrease in trait 2. What do I actually see? Remember, both traits are heritable, heritable, heritable. I get no response in either trait. Why? Look at the determinant of this matrix. 400 minus, this has got a determinant of 0, and it turns out that's the eigenvector corresponding to 0. So in that direction, if you imagine a scatter plot of all your values, that scatter plot is actually a line. So I can move up and down that line, but I can't move at right angles to it. There's no width in it, basically. This turns out I just picked it to be a vector at right angles to that line. So there's no, even though I can change trait one by itself and I can change trait two by itself, I can't change them in this particular combination. So in this case, I'd get no response. And that's an example of an extreme genetic constraint. So the key for the second part of multi-trait selection is the multivariate Breeder's equation. R equals G P inverse S allows you to break up the actual target selection. P inverse S gives you a list vector beta, the target selection. G gives you the amount of rotation that you can see on that. Okay. Again, a lot of details. Don't get hung up on them. Think about the basic idea. The basic idea here is you cannot predict single trait. You cannot predict multiple trait response by just using a bunch of single traits. You've got to not use the heritability. You've got to use the whole P and G matrices because heritability ignores phenotypic and genetic correlations, whereas the G and P matrices take those into account. So that's a quick overview of multiple trait selection in a broad sense. I'm now going to pivot over and talk about index selection. But any questions before I do that? So index selection basically has, it's got two parts. Back up a second here. Chapter 37 you have is the theory, and chapter 38 is applications. There's a huge amount of detail here, which is technical. So I'm going to go pretty quickly because I want to give you guys the feel without getting bogged down in the details. So you'll see a lot of matrix expressions here. Don't get freaked about them. That's something you look up later on. I want you to get kind of the idea about what you're doing. So practically, when you have multiple traits, how do you select? Well, let's look about independent culling. Independent culling means I go out and I measure trait one, and I only pick plants that are above a threshold in trait one. Then in that group, I then measure trait two, and only those that are above the threshold in both one and two are saved. That's called independent culling. Another option is if you're either above a threshold in either one or two, I take you. Independent culling might make sense. For example, you screen for disease resistance early on, then screen for yield later on. Index selection says that I choose individuals based upon a weighted index. So 2 times trait 1 plus 3 times trait 2. Index selection turns out to generally be the most efficient way to select for multiple traits. And we're going to then flush out this idea about index selection. But this just reminds you there are other ways that you can select on traits besides using an index. What an index does is it takes a vector of measurements and it turns it into a single value for each individual. For example, a nutritional score. You have a bunch of grains in front of you. A nutritionist basically gives you a quality score based upon points for certain things and deductions for others, maybe lipid content or certain types of protein. So then each of your strains then gets a single nutritional score that's based on a lot of different characters. If you choose the strains based on that nutritional score, that's selecting on an index. So chapter 37 gives you the basic idea. Now, just a couple of quick things to help you kind of read these expressions. So if you see B transpose Z, that's a weighted sum. That's a scalar. B transpose PB, that's a scalar. As we saw in the last lecture, that represents the variance of this weighted sum. So weighted sum, it's variance. But now if you see GB or PB, that's a column vector, right? So the reason it's a scalar here is I've got a 1 by n in front of it. So I get a 1 by 1. These are column vectors. So these will give us within generation changes. These will give us how the traits change, a vector of how the traits change. These will give us information about how the index changes. So what exactly is an index? Well, I have weights. Not going to worry how we get those weights for now. And then each trait is assigned a weight. So I go and I give you a total score based upon your total index value. And that's your index. I've now taken a multivariate vector of data and collapsed it to a single number. And selection on an index is really easy. You take all your traits, you make the index, and you write that on a card and hold it up. That's your index score. You simply go and pick individuals with the highest index score. That's it. What's the phenotypic variance at index? It's given by this quadratic product, the genetic variance by that quadratic product. What's the heritability? Well, it's the genetic variance divided by the total variance. So that's the heritability. I can now talk about what's the response in that index. And the response at index is simply given by here. It's the heritability, square root, times the square root of the added variance times the intensity. Remember, the intensity is what fraction you save. You save 5%, the intensity is about 2. You save 20%, the intensity is about 0.7. So we can then use this then to get an equation for the response. So before we get that, that's the data we saw before with soybeans. Here's our weights, 2, minus 3, 4. And from those, that is the phenotypic variance, B transpose PB. That's the genetic variance, B transpose G. So the heritability, the ratio of genetic to phenotypic is about 0.4. So this index, 2 oil minus 3 protein plus 4 yield, that index has a heritability of about 0.4. And the response is just simply plugging things in here. This is one way to write the response. That index is another way to write the Bridges equation. We know what those are. Remember, that's a scalar. It's a number. That's a number. So I can write it this way. So that's a number, and that's a response in that index. Now, just because an index changes, it may not be what we want, because index changed by the components changing. And some components may change in a direction we don't want them to change. So how do we figure out how the components change? We can find out basically, if I give you the weights, you compute an S vector. You can take that S vector and plug it in the multivariate Bridges equation. And this here, that expression, gives me a vector of how component 1 changes, component 2 changes, component 3 changes. So let's look at this. See, it says G beta. Let's look over here. This is the exact same expression. Now, beta transpose. What does that mean? It means I take a weighted sum, B1 times the first component plus B2 times the second component plus B3 times the third. That's the change in the index when I do that. And this expression over here is how the individual components change. So in index selection, we're concerned about how the index change. We also know how the individual traits change. And going back to our example over here, from oil, if I save the upper 5%, my index response is going to increase by about 207. So if I just pick individuals based upon that index weight, in the next generation, that index score is going to increase by 207. Now, that's a good or a bad thing depending upon what the components do. And there's no right or wrong answer. We can look at the response in each trait. So what we see is that's the response in the 3 traits. And so how do I get the 207? 2 times that minus 3 times that plus 4 times that gives me 207. Here are the actual targets of selection. And you see they actually match up because these are proportionally the same. What we've done here is we've called selecting on a base index. That's how I want the traits. That's what I'm going to be scoring. I simply make an index using those weights and then pick individuals based upon that. But we can do better. So recall we saw earlier this notion of a indirect response can be larger than direct response. This is called the Smith-Hazel index. And here's the idea. I want to move in this direction beta. That's where I want to go. That's my weights. When I multiply beta by G, though, I rotate off that. And that's my actual response. Is there some way to select so when I rotate, I get back to where beta is? And those are called Smith-Hazel weights. So for example, if A are the weights I'm trying to improve, if I pick individuals based upon that weights, I get over here. However, if A is the direction I'm trying to improve, can I choose weights B such when I rotate them by selection, I end up along the direction? And the answer is you can. Those are called the Smith-Hazel weights. And basically, here's what they look like. They look like this. So they depend upon P inverse and they depend upon G. And so in the Smith-Hazel weights, I pick individuals. So A is the index I'm trying to maximize. Instead of selecting directly on A, though, I use these additional weights, which account for genetic and phenotypic correlations to give me a larger response. So in particular, what are the Smith-Hazel weights? Well, here's the index I'm trying to improve. Here are the Smith-Hazel weights. So much less weight on yield. And when I use those Smith-Hazel weights, the response I get is 248. So instead of 207, I get a response of 200 and basically 50. I get a 15% increase. Again, A are the weights I'm trying to improve. That's what I look at in my offspring. But if I pick parents based upon these Smith-Hazel weights, B, given by here, so they're the weights I'm trying to improve, I need P and G, I get a larger response. Instead of 207, I get 250. So this is called the Smith-Hazel index. In most cases where you see a selection index, people have used Smith-Hazel. So the idea here is I could pick individuals based upon these weights. If I do, then if I look at the response in this index and the offspring is 207. But if I pick parents based upon these weights, the main difference here is in yield. The response in this index and the offspring is 250. So a 15% larger response using Smith-Hazel. Smith-Hazel is great in theory, in practice, because it depends upon all these genetic and phenotypic correlations, especially genetic correlations. It's hard to measure them accurately. If you use inaccurate rates, you can get a worse decision. So people can either use what's called an estimated index. You estimate P, that's pretty straightforward. G's got a lot of error in it. G's can be hard to estimate. And the general rule of thumb is with 3 or more traits, it's hard to get a good estimate of G. Why? Because you have a whole bunch of covariances. So that's an estimated index. If I knew P and G exactly, Smith-Hazel would be best. If I estimate them, there could be problems. A base index means I just choose individuals based upon the weights I want, not any corrections. Heritability index splits the difference. It says pick the weights based upon the actual weights you're going to improve and the heritability. Traits with higher heritability get more weight. Why? Because their phenotypes are a better predictor. And you see these two indices are the same when P and G are identity matrices. If you have no genetic and no phenotypic correlations, Smith-Hazel gives you the heritability index. So up to this point, the key idea for a selection index is this. And again, there's details which you can look up. Here's the basic idea. I'm trying to select on a whole bunch of traits. So I come up with some model in my mind about how important they are, some economic score, some nutritional score, and I then make a weighted index. So when I measure you, I take your individual trait values and multiply them each by a weight, and I sum that. That's your index score. Then I simply select you based on your index score. I gave you expressions to tell you what the heritability is, how the components change. A clever trick you can do is if you've got genetic and or phenotypic correlations, you can do better. If I'm trying to have individuals with a certain index, have that index increase in the offspring, if I select parents based upon different weights, I can actually get a larger response in their offspring of my original weights. And those weights are called Smith-Hazel. They're chosen basically to account for and adjust for the effects of the correlations. So key idea is, what is an index? When you select on an index, you change the index value. You also change the components. You now have equations in front of you that tell you how to do both of those. You also have Smith-Hazel weights, which tell you how to compute those optimal weights if you have good precision about what P and G are. We're now, after questions, going to go on and ask the final part, which is about what's called the restricted index. And let me pull chat here. And in a restricted index, come on, chat, you can come up. Be good for me. My chat's not coming up here. Um, in a restricted index, you may not want certain components to change. So questions about, so index selection is what you're going to do when you do genomic prediction. It is a type of index selection. You basically have a whole bunch of SNPs, you get some weighted score of the SNPs, and you use that then to get your genomic predictor. Okay.
So Bruce, there
Yes.
is one question on the chat from
Yeah,
Johannes.
I'm not, my chat's not opening for, oh, there's the chat. Okay. Yeah, I'm gonna hide
No worries,
myself.
no worries. If Johannes is in the room, and you're happy to unmute, you can speak to your question live if you'd like.
Yeah, so I see basically the question has is an excellent question. It has to do when you have a real world experimental design, how do you account for all of these things? And we mentioned this briefly a couple of days ago. So what you do in your real world design is you would get either blue estimates or bluff estimates of your genotypic values. Blue estimates, if you use basically treat them as fixed effects. Bluff estimates, you treat them as random effects. The advantage with random effects, you can borrow information from relatives. And then those are what you would plug into that design. We'll talk about that a little bit more when we talk about Bluff and Remmel a little bit later on in the series of modules. As terms of R packages, there are better people than me to make an estimate on that. Is Nelson in here? He might have a better idea. I don't see him. Oh, there he is.
Yeah.
Put on your Arsenal jersey, buddy.
So sorry, what was the question again?
So if you look in chat, basically it's about what R program is best to estimate genetic and phenotypic variances in a randomized complete block design.
I use SREM or R in most cases.
So Nelson is one of our instructors, and I would recommend just get in touch with him because that's more in his area than my area.
Yeah, I can help you. Mostly I use SREM or R to kind of construct the genetic and phenotypic variances, but there are so many other softwares you can use. Just that SREM or R is kind of like you have to subscribe to the software, like a paid version, but I can show you how to use other freely available softwares online, yeah.
Yeah, so Nelson's great at that. And I'd catch him soon because with England coming up on Saturday, he could be a real mess. So we'll play that by ear. Other questions? So let me finish up then by talking about the idea of restricted indices. So suppose you have a scenario where you have a trait that you cannot change, period. Well, you don't use an index because an index is going to drag that trait away. You might go, I'll put a heavy weight on it. That doesn't matter too much. Or even worse, I'll put no weight on it. So restricted and desired gate indices are now you're trying to manipulate how certain specific components themselves change. In particular, two scenarios, you don't want a couple of traits to change at all. You want to maximize the response in the others, or you want to specify the exact proportions how people change, how traits change. When you do either of those, the net result for that improved control is less total response. So suppose in the soybean data, you want no response in trait 1, but you want to maximize response in trait 2 and trait 3. So if you use Smith-Hazel weights and you say, well, I don't want trait 1 to change, so I'll put no weight on it, that's the worst thing you can do. It means you don't care what its value is. And what you actually see is the response in traits 2 plus 3 is 37, but trait 1 changes by 6. And that may be catastrophic for your particular program. So is there a way to force that trait not to change while maximizing the rate of change in the other? And the answer is yes, and those are called Smith-Hazel weights. And the classic example of that is you've got two correlated traits, not Smith-Hazel, those are the Kempthorne restricted index traits. Sorry, it's early in the morning for me here. So the classic example is I've got two traits. Trait 1 is great where it is. I don't want it to change. I want to change trait 2, but trait 1 and trait 2 are genetically correlated. So Here we are. So let me reverse the order. I want to change trait one. I want to have trait two not change. Matrix multiplication, those are the conditions. Those are the weights. So if I put those weights, it turned out to be this way. If I pick individuals based upon an index of trait one minus this weight on trait two, and what you see if trait one and trait two are not genetically correlated, that weight is 0. If I pick individuals based upon these weights, I get trait one changing and trait two not changing. That's an original two-trait restricted index. The more general version, due to Oscar Kempthorne, it looks complicated. There are the Smith-Hazel weights. There's some stuff in there. Again, the notes will talk with you about that. You can go ahead and do those when you compute those weights. So remember, what this does is it simply gives you a vector of weights. It's hard, they're tricky to get to, but then you select on those weights. When you select on those weights, which are these, the response you see is, hey, that's great. There's no change in trait one. Perfect. But the total change in trait 2 and 3 is only 15, whereas over here, the change was 37. So when you put a restriction on there, it comes at a cost. And you have to ask yourself, is no change in trait 1 and 15 in 2 plus 3, is that better than 37 in 2 plus 3, but a 6 in trait 1? There's no right answer. That depends upon your breeding program, and it could vary over those. So that's the example of restricted index. You force certain traits not to change. And this basically says trait one doesn't change. You can elaborate much more generally. And by the way, it doesn't matter what weight you put on here, because putting the one there will ignore that if you look at this matrix expression. Again, the notes will cover that. You can do other things. You can put multiple restrictions on there. That's a Talus index. But the other one people use is something called a desired gains index. And this seems appealing. My desired gains are equal changes in traits 1, 2, and 3. Well, those weights are easy to figure out, right? That's R is the response. I want it to be proportional to 1, 1, 1. Then if the weights are simply G inverse times my desired response, and I standardize the weights. By the way, what matters on the weights are the relative proportions, not their actual numbers. You can always standardize, so 1 is the value of 1. So you put those weights on here, and then use those weights to choose individuals. That's the response you get. So what would happen if I would put desired gains on? So we have, remember my scenario before, we don't want oil to change. We want to maximize protein plus yield. If I use Smith-Hazel weights, my response in what I want is 37, but I get a big response in trait 1. If I use a restricted index, so I'm trying to maximize this sum and have trait 1 not change, I get a response in 15. If I use desired, I want no response in trait 1 and equal response in trait 2 or trait 3. I can get that as well, but now my response is 6. So as you add more constraints, the net result is the response in the part of the index you care about goes down. There's no free lunch. There's a cost to using those constraints. So the key idea about desired gains and restricted indices are with normal selection indices, I simply want to maximize that weighted sum. I don't care how I do it. With desired gains, there's a part of the sum I want to maximize, and the other I don't want to change at all. So I can figure out what the weights I have to choose individuals are to get that result. That has the advantage of those components you want not to change won't change, has a disadvantage of those things you want to maximize not being as large. The other extreme is desired gains, where you specify the relative proportions of change. That's the worst case scenario in terms of maximizing the response and things you want. So as you add more restrictions, you end up having more issues. And I think I'll leave it at that. There's a couple of applications, but you can go through those on your own. So summarizing multiple trait selection, we've got genetic and phenotypic correlations. You can estimate a genetic correlation by estimating a genetic covariance by simply measuring trait X in one individual and trait Y in a relative, and using expressions to connect those two. The We talked about a direct response on a character and a correlated response. We then talked about the multivariate breeders equation, where you now have to plug in instead of heritability. So with 5 traits, instead of having just 5 heritabilities, you have to plug in all the phenotypic and genetic covariances as well. If you don't incorporate those, you'll get an erroneous result. Then we talked about one way to select on traits is to take your traits and make a weighted index. That's called index selection. Each individual then has a single index score. It makes it a single trait problem. You then pick out individuals with the highest index score. We gave you expressions for what the response is going to be and how the individual components in that index are going to change. Then we talked about doing slightly better something called Smith-Hazel weights. I can select on individuals based upon the weights I want them to improve on, or I can do a special trick and use slightly different weights to pick parents. When I pick parents using those different weights, I get a larger response in that index I want than if I pick parents based upon that index that I want. It sounds like magic. It's basically borrowing the phenotypic variances and covariances and using them. Then we talked about trying to control how individual elements that index change. With restricted index selection, I'm forcing one or more traits not to change. Then I'm doing Smith-Hazel to maximize the response in the others. So I have an index I want to maximize the response in while holding other components constant. And finally, desired gains, I actually tell you the specific proportions I want for each. That's the worst of all of them in terms of the actual response, but it gives you the most control. And that's your kind of overall introduction to index selection. Right after here, I will also send the chapter on to post on correlated characters, which talks a little about some of this material as well. Not as much as the chapters I gave you, but it'll talk a bit more about estimating genetic correlations and about some of the G by E aspects. Okay. So questions or comments about that? A lot of stuff. Again, a lot of details. I try to go through the details fairly quickly because I want you to get the main concepts. The details are what you look up. Main concept, when you've got 2 or more traits, you can't use the breeder's equation. Single trait. You've got to use something else that accounts for the phenotypic variances and covariances. Practically speaking, when I select on multiple traits, I'm selecting on an index. We talked about how to estimate what the response to that index is going to be, what the response to the components are going to be, and then ways to manipulate how I weight individuals to control how certain of those components change or don't change. And those are the key ideas. Patrick, I love your eagle. I appreciate it. Any other questions? Yeah, so Nelson presenting in module 3 along with Alex. Hi, Bruce. Nelson. Okay, let me. Sorry, so you don't see me doing this because I got my screen's really small. Turn off my thing. Nelson's. If we already have GEBVs for multiple traits, yes, multiply by the economic weights. Period. You've already done that. When you get the GEBVs, you've already removed all the correlations. You can just do it directly. You're home free. Okay. Well, it's been a pleasure having this. Zoom is not nearly as fun as in person, but it's been a pleasure interacting with you. I look forward to doing more of this in the future, and also hope forward to interacting with some of you directly for those subset of people that are going to be in Cape Town in the fall. We have a question. Yes.
Hi, I just wanted to note that there are just 2 more questions that
Oh yeah,
came
no problem.
through
Sure.
on the chat. I can read them to you if you'd like.
Okay, hang on. Let me just again, so you don't see my bald head shining on you here.
No problem.
Come on now. There we go. Okay. Um, can changes in response to multi-trait selection assist you in informing the weights? Yes. So a big thing we didn't talk about is what are the weights? And there's no right or wrong answer to that. The weights are you as a breeder, what you think is desirable. When you're breeding, you often write down a product description. And the way I would recommend you doing this, should I use this or weighted index, is run the math and see, okay, if I use a normal index, here's my expected response. If you use a restricted index, here's the response I get. From a breeding standpoint, which makes more sense to me as a final product? There's no right answer. It's something that you as a breeder have to do. It's why Chat GPT won't replace you as a breeder, for at least a couple more years. The other question Uh, yeah, so Samar is talking about that. Paul says often our data is not very accurate. If the data, that's a great question. If data is not very accurate, do not use Smith-Hazel. If data is not very accurate, use the base index or the heritability index. Heritability kind of splits the difference. If you've got some really lowly heritable traits, I'd use the heritability index. But again, if you don't have a lot of accuracy, then Smith-Hazel is challenging, but also desired gains and restricted is also very challenging. Okay. And Sumar has also commented on that. I believe that all the comments in here are also going to be available to students in that. So any final questions? Well, thank you all. Like I said, I'll forward that chapter on correlated characters in the next minute or so to be posted on the website, and look forward to seeing you soon. Okay. Good luck, everyone. Cheers.
Thanks very much, Bruce.
Okay. Bye, Bruce.
Bye-bye. Cheers.
Thanks to everyone for joining. We'll see you at the next one.